Good morning, everyone. My name is Bill Wells. I'm coordinator for this class, for this presentation. And we've been working on this for almost 2 years. And originally it was going to be presented by some of the students at Hope College. But the ones that were originally proposing to present have actually graduated and moved on now. So the professors are left to do the dirty work for them. So the presenters are K. Greg Murray. He's currently the T. Elliott Weir Professor of Plant Science at Hope. His BS and MS in biology are from California State University, and his PhD in zoology is from the University of Florida. He joined Hope College Biology Department in 1986. The other presenter is Peter L. Gontier. He's Professor of Physics at Hope. His BS and PhD are from Texas A&M University in chemistry and nuclear chemistry. Since 1992, he has been pursuing the astrophysics of neutron stars through modeling and microphysics in strong magnetic fields. So I'm not sure with that background what he's doing underwater for this presentation, but we're about to find out. So I will shut up and let the presenters proceed.
Hi, everybody. I am going to, I'm Greg. It's nice to see so many people here this morning. I'm going to share my screen, and it'll take me a second to get this going because I'm using two monitors. Let's see here. Well, bear with me just a second because I got to get this correct. Okay, you should be seeing the title slide now. So Dr. Gontier and I are going to sort of switch off here. I'm going to go first, and then he will go, and it may come back to me a little bit later. But anyway, we're going to talk about the course that we teach. We're not going to talk about just the part of it that is done in Cozumel, because this course sort of is something that probably could only happen at a place like Hope College, where people like an astrophysicist and a tropical plant ecologist meet each other. That would never happen in a big university, probably. And so we want to talk a little bit about the structure of the course and stuff like that, and we'll show some pictures and so forth. Just an overview of the course. The course is cross-listed as a biology and biophysics course, or a physics course. And it's really, this is really kind of from a catalog description. It's an exploration of the intersection of physics and marine biology. We are really interested in how the natural world works, and we like the unity of the natural sciences. And so with that interdisciplinary sort of interest, we wanted to explore how physics informs an understanding of marine organisms and marine environments, and talk about sorry, you might hear my dogs barking in the background sometime. We wanted to explore how organisms have dealt with the physical challenges imposed by the environment. And so the prerequisites for this, for most people, are just introductory biology, which at Hope College right now is 2 courses. The genesis of the course, I'm going to talk about that in a little bit of detail here because it's kind of fun for us. Just comes through, serendipity, and then an incentive that was offered by the college, and then that shared geekiness that I'm referring to, which, Peter and I would prefer the term shared passion. But years ago, our dean of the natural sciences, Jim Gentile, used to host lunches for members of our division, excuse me, who were interested in talking about the GEMS program. The GEMS program stands for General Education in Math and Sciences, and the courses in that program are mostly targeted towards non-science majors. the way the program was developed, a large proportion of the courses that students would take to satisfy their science requirement had to be interdisciplinary. Anyway, my wife and fellow biology professor, Dr. Kathy Winnett Murray, was sitting next to Pete at the lunch, and as they chatted, he mentioned that he was interested in marine biology and that he was a certified scuba diving instructor. And so Kathy said, "Oh, well, you know, Greg used to study marine biology when he was an undergraduate. I was headed in that direction until I veered off into marine birds because of Kathy." And then she said, "Oh, we should get together and talk about our common interest." And we found out that we had a shared passion for understanding how the natural world works, and saw the boundaries between the different sciences as kind of artificial, more as necessities for individuals to specialize in order to make new discoveries than as fundamentally different disciplines. And so we really like the idea of the unity of the natural sciences. And so a few years later, the theme of the college's Howard Hughes Medical Institute grant was interdisciplinary science. And there was little pots of money for people to get together across departments and develop courses that were truly interdisciplinary, that would be developed and taught by people from different departments. And so we pursued it with that. And just with an idea towards teaching a course that was kind of like the traditional discipline of marine biology, but veering off frequently to explore how a physicist would approach those kinds of questions and problems. So we kind of put all that together and developed the course. And the way it evolved is that it has 3 traditional lecture periods per week, 1 3-hour lab per week, so very typical for most science courses. The lab, the first half of the semester really consists of a survey of the major groups of marine organisms through examination of specimens and some dissection and stuff like that. And then the latter part of the semester is really based on more inquiry-based experimental labs on the physics of water and marine organisms and stuff like that. And we'll talk about some examples of those things. The students have the option of getting certified to scuba dive. And for the first couple of years that we did the course, Peter was the instructor himself for that part of it, and it took place outside of class time. But later on, we actually had instructors from a local dive shop do that part of it for us, because it was just, you know, harder for Peter to keep up with all of that. So the students who wanted to get certified to scuba dive did so outside of class time. Some years they actually got PE credit for that because there was a course on the books at Hope College. And then those were the students that went on an optional spring break trip to Cozumel that we'll talk about in more detail. So not all of the students went to Cozumel. Yeah, so there's the optional spring break trip, and we'll talk about that in more detail in a minute. So for the first part of the semester in the laboratory part of the course, we concentrate on surveying the major groups of marine organisms. Hope has a pretty decent collection of marine invertebrates that was mostly established by Dr. Alan Brady during his years of teaching the Florida May term, and later Dr. Tom Bultman, who's still on the faculty. We also have pretty extensive collections of fish, mostly established by Dr. Eldon Gray and others like Dr. Brady. Most of our fish are actually freshwater species, but some of them are marine, and even the freshwater ones are members of groups that also have representatives in marine systems. So they're still useful for teaching about the characteristics of those different taxonomic groups. We also have a pretty nice collection of some more of the fish there and closer up. We also have a nice collection of birds that was mostly established by Dr. Gray over his years on the faculty. I would just like to take just a minute to point out that most of these specimens are really old, and we're no longer really collecting them very much. We do some collection, but But we really take care of these collections. But it's really a labor of love over the years of people like Dr. Gray and Dr. Brady and Boltman, to some extent myself, but also largely Dr. Kathy Winnett Murray, who teaches the vertebrate zoology course. Maintaining these specimens is a fair bit of work and requires care if the students are going to be using them without them getting degraded over time. And so without collections like this, there's literally no way for a college student to have any sort of hands-on learning with the great majority of living things on this planet. Classes that use collections in this way used to be taken by most students that were pursuing majors in biology at any university or college in the country. But with the changes in the curricula, which have been necessary because of the expansion of knowledge and so forth, and we only still have 4 years to pursue a degree. Nowadays, only a handful of students graduating even with a degree in biology have any connection to these kinds of organisms at all. So it's a real special thing. We also have, the college does not have a very good collection of marine algae, but I have a bunch of specimens that I collected while I was a student taking a marine algae course back in California. And so I use some of those for specimens that we use in the class too. So that's a kind of a fun thing that I established over a long time and just kept over the years. So the labs that we do that are more inquiry-based or more experimental are of a variety of sorts. And I'm going to give a couple of examples of these, and then Dr. Gonthier will go into more detail about another one in a little while. One example of these was to measure the flexibility of wave-swept algae. We put these in the second half of the semester because we want the students to have the experience of that survey of all those different kinds of marine organisms early in the semester before the ones that are going to Cozumel go there. But later on, we do these other kinds of labs primarily. But here's one example. It just concerns the biomechanical properties of marine algae. In these kinds of habitats that are found along the shorelines of much of the world's oceans, these are really stressful environments. And in these wave-swept environments, to survive there, you either have to be attached flat against the rocks really firmly, but if you're not, then you're going to get whipped back and forth, and you have to be really flexible, and thousands of times a day, you're going to be pounded by these waves. And so it's a really physically stressful environment to live in. And so we actually use, oops, the right part of the screen here. Here's an example. This is called feather boa kelp. This is from Southern California, where I'm from originally. And it's one of those things that gets whipped back and forth, but lives there for a long period of time. We actually use an instrument called an instron in the engineering department to measure the flexibility of these things. Engineers use that machine to test designs of mechanical devices like bridges to see how much force you can apply to them before they fail. But we use it to estimate how much different species of algae can bend before they break. And it turns out that these things are more flexible than just about anything that human beings can manufacture. It's really difficult to find a human engineered material that can withstand that kind of wave stress. So anyway, this is one of the things, and I'm not going to show the math associated with the measurements that we take, but we use that in just the same way that an engineer or a physicist would use. This is just the algae that's attached to the jaws of the machine. A bunch of the labs that we do. Well, let me break right there and see if there are any questions that people want to present. Do you have any questions?
Nothing yet.
Okay. So we spend quite a bit of time in the lecture part of the course talking about the physics of light in marine environments, because of course, all living things depend on photosynthesis to one degree or another, sometimes directly, sometimes indirectly. But light gets scattered and absorbed pretty quickly as it passes through water. So there's something there that terrestrial organisms don't have to deal with. But it turns out that different wavelengths of light, different colors, get absorbed differently. The red wavelengths get absorbed more strongly than those toward the blue end of the spectrum. And so blue light penetrates into the water much deeper than red light. This is just a diagram from one of the textbooks that we use, just sort of showing that the blue end of the spectrum penetrates better than the red. Well, we use an instrument called a spectroradiometer to measure this in class, but I wanted to point out this diagram also from a textbook that just shows the absorption spectra of two of the variants of chlorophyll. And of course, this is the photosynthetic pigment that all organisms that can photosynthesize use. And chlorophyll A, which is the common one, it absorbs in this part of the spectrum, which is in the blue, and also in this portion in the red. Chlorophyll doesn't absorb the green and yellow wavelengths, and that's of course why green leaves look green. It's because that portion of the spectrum is not absorbed. But the instrument that we use is down here. It's called a spectroradiometer, and we can use this to pass light through a 1 meter long column that we can put anything in, any liquid or, well, any fluid really into. And what we can do is we can measure at 2 nanometer increments what the intensity of the light is reaching the sensor, which is down here. And then we can record and analyze that. And so we can fill this column with air, or we can fill it with water of different sorts and measure how well the light gets transmitted through it. Here's just one of the sample data sets that we collected here. And what you can see is that it's actually the red wavelengths, those that are around 700 nanometers here, well, 650 to 700 or so, gets reduced the most by water. This line here is for the air. And so this is the intensity in watts per meter squared of the light. This is in air. The green line here is for fresh water. This one here is for salt water, and this one down here is for fresh water into which we have a suspension of photosynthetic plankton cells. And so you can see how much the light gets filtered out, even by a meter, which is about 39 inches of passage through a fluid, each of these fluids. But the reduction from the intensity in the air is greatest at these red wavelengths. It's less at the blue wavelengths down here. So if you're a plant, it's going to be hard to live at depth because very little of the red gets through, yet red is one of the wavelengths that the plants are absorbing with chlorophyll. So Pete came up with a way for the students who went to Cozumel to experience the effects of depth on wavelengths of light available with their own eyes. And so what he did was he made a bunch of these little slates that have colored beads and colored squares of plastic on them. And then we had all of the students take those slates with them while they were scuba diving and to look at the colors from time to time when they were at different depths. You'll note in a lot of the photos that we show later today that everything looks bluish, and this is why. We're not using it, it's because we're not using flash. But when we do use artificial flash, you'll see that there are lots and lots of vivid colors even at depth. But here's what those beads look like at the surface when they haven't passed through any water at all. This is a set of pictures that Peter made with a depth gauge next to them. At just 11 feet, look at the red one and the red bead here. It's almost black. You can barely see that it's red. And the reason is the red wavelengths of light are filtered out so quickly that they're not even reaching, most of the red is not even reaching 11 feet. When you go down a little bit deeper, so this is 33 feet, there's no color, well, to my eye at least, there's no color here left at all. And you're starting to lose the intensity of the other colors as well. This one is from 66 feet, and you can see all the colors are becoming more subdued. These beads here are a little bit different because some of these are actually absorbing ultraviolet light and then fluorescing in the visible. So that's, we didn't know that before we put them on these ones. So look mostly at the squares here. And then by the time you get to 95 feet, you've lost all the red and you're starting to lose the yellow as well. And but the blue is still there. And it's sort of a demonstration that students could see while they're in the water themselves. We follow up on that, some more on the physics of light and water, because people have noted that certain kinds of marine algae, like these giant kelp off the coast of Southern California, can grow at depths of 100 meters or more, and yet others can only grow really close to the surface. And so the reason for that is that some groups of algae have evolved accessory pigments that serve to capture light at the wavelengths that penetrate to greater depths and then pass along that absorbed energy to chlorophyll, where it goes into the regular photosynthetic pathway. So we order a bunch of species of, or several different kinds of seaweeds from a biological supply house on the West Coast. And then we have students make extracts of the pigments from them. And then they measure the ability of those different extracts to absorb light at different wavelengths in a spectrophotometer. And so this is in a molecular biology lab, actually, upstairs in the science center on campus. And so we're, the student is measuring the absorbance of those pigment extracts. And so here's a data set from one of those. And the way you read this here, this is wavelength in nanometers. And so the red is down here, the blue is over here, and this is absorbance. So it's not the light that's transmitted through, it's actually the absorbance of each of these pigments. And these are just from some different species. Ulva is a species of green algae that only lives close to the surface. You never find it at depth more than maybe 8 or 10 meters, not even that much, probably only 5 meters or so. Egregia is that feather boa kelp, so that's a brown alga that lives at greater depths, and Gigertina is a red alga that can grow much deeper. Spirulina is actually a blue-green alga here too, but it has some of the same pigments as one of these other ones. Anyway, what you can see here is that ulva doesn't absorb well. I mean, they all absorb in the red okay, but the ulva doesn't have any of those accessory pigments that can absorb the blue light even better, and even the violet light as it penetrates deeper in the water. But Egregia and especially Gigertina do. The reason why they can grow deeper is because they have these accessory pigments that are not chlorophyll at all, but accessory pigments that absorb the shorter wavelengths really effectively and pass the energy on to chlorophyll, and then it goes into the regular photosynthetic pathway. Here's another example of one of the labs that we do. This one we actually do in Cozumel. This is not a picture from Cozumel, but I use this picture because it shows the intertidal environment really well. And so we examine the thermal biology of intertidal organisms and look at the effect of shell color and microhabitat. This is a stressful environment for a whole bunch of reasons. One is because of wave stress, of course, but the other is because it's a thermally stressful environment. If you're in the tropics especially, it can get really hot during the middle of the day at low tide, and yet a lot of organisms do live in these intertidal habitats. You have to be able to be attached to the rock where you can't move very far. and despite the fact that the rocks get hot enough to fry an egg in a lot of cases. So that's an interesting phenomenon to which the understanding of physics of heat transfer and stuff can contribute. So we examine this with an experimental an approach that we developed ourselves. You can see that this is just some barnacles that are firmly attached to the rock, and these are snails that can move around some, and you can see they're congregated here in the crack, and there's a reason for that, and it has to do with why we did this lab. We took a bunch of shells, empty shells of marine snails, and we can examine or investigate the consequences of different shell sizes and shapes and textures on the way at which they, or the rate at which they heat and cool at different times of the day. And we can even paint some of them. So the data I'm going to show are actually from these two shells here, which are moon snails, but we've painted one of them white and one of them black. And then we put little thermocouple thermometer probes into these after we fill them with a gel that has similar properties to living tissue. And here's a data set that we collected, and this is late afternoon. We did this after a full day of diving in Cozumel. And what I'm showing here is starting at 4 o'clock in the afternoon and going to 4:45, measuring the temperature of the air, which is these diamonds here, and the water temperature, and this is in degrees Celsius, of course. And you can see those don't change much over time, but pretty quickly, the white shell in yellow here and the black shell even more so gain a lot of heat. Now, if this was around noon, we'd find these temperatures going up well over 40 or up to 50 degrees. The temperature inside the shell can get up to 50 degrees Celsius. That's about 122 degrees Fahrenheit. And these critters can't pant or sweat, so they have to have other adaptations to avoid what would otherwise be lethal temperatures. And so there are a bunch of those that we talk about as a result of this too. Okay, so that's as much as I'm going to say on these labs. I want to talk a little bit about the Cozumel trip, and Peter's going to show a video of some of that in a couple of minutes. But besides scuba diving, what else happens on Cozumel? Well, I want to say a little bit about that. In the afternoons and the evenings, before and during dinner, we usually pass the field guides around and agree on which species we saw during our dives that day. We always compile a list of species that we were able to identify over the week. This is Dr. Gonthier's son and Hope College graduate in biology, and now he's a professor himself. This is Dave Gonthier. Some of you might have met him along the way. But anyway, we usually go out to dinner as a group. As you can see, we try to go to more out-of-the-way places away from the waterfront because they're not frequented as much by tourists from the cruise ships, and they're thus less expensive. And so Peter knows these places really well since he's been going to Cozumel so many times over the years and speaks Spanish fluently. In fact, he knows just about, I haven't found anybody on Cozumel that he doesn't know personally. So he's a great resource for that. And so the students really appreciate paying less for dinner anyway. So we have a good time on dinner, and the students get to experience some of the culture a little bit better as well. The thing that we do on the last day when we're in Cozumel is a little bit different too, because it turns out that you can't fly safely for at least 24 hours after your last scuba dive without putting yourself at risk for decompression sickness, what people call the bends. So on the last day before we fly back, we always rent a couple of vans, depending on the number of students we have, and we take a tour around the island to see some terrestrial habitats and the organisms there, because otherwise we would just be killing time around the pool, and that's not compatible with, our reputation as brutal taskmasters. So this is Cozumel, and the town is right here, but we take the vans around and tour the island at pretty much every place that you can drive. and experience the different habitats there. One of the places that's a really great resource for us is a park down at the southern end of the island, where there's a lighthouse and a nice park that has wading birds and stuff. And so we go up in the lighthouse and people can see the lay of the land there. These are some of the students there. That's what it looks like from up at the light itself. And these are really cool, close marine habitats. And of course, we get to do some botanizing while we're there. And here's a warbler, a migrant warbler from North America that's wintering there, because we usually go in March. And then we spend time in this estuary looking at wading birds. And these, by the way, are, for those of you who are birders, these are roseate spoonbills, really cool birds that you don't see here in the United States very much. But there are also some of the birds that we know from here, like great blue herons. And there are American crocodiles there. Now, everybody's seen alligators, but not many people have gotten to see American crocodiles, but they're really common there. So that's what we do the last day, and then we fly the very next day. The last thing I want to say before handing it over to Peter here is about the list that we compile every year. Every year we take a lot of trouble to identify the species that we can from using the field guides, the print field guides that we take with us. And every year we have, that list has about 200 to 250 different species on it. Those are the ones that we can just identify to species. There are many times that many that we see, but we just can't identify them as precisely. And that's really, in my view especially, that's really the most important benefit of this whole trip to Cozumel. Giving students the chance to see a huge diversity of living things that fewer and fewer people ever get to experience, even biologists. The best estimates are that about 25% of all species in the ocean are found on coral reef habitats, despite the fact that coral reefs cover only about 1% of the world ocean's total area. Moreover, 8 of the 34 or so phyla of animals on this planet. Now, phyla are major groupings just behind kingdoms. So it's like all the chordates, all of the arthropods, all of the mollusks. Those are examples of phyla. 8 of the 34 of those animal phyla on the planet are only found in the ocean. They have no representatives on land or in freshwater ecosystems. And so bringing students into contact with so much biological diversity is the thing that really sticks out to me as being the greatest benefit of taking students to Cozumel, because it really is astounding. It's one of the, people have called coral reefs the tropical rainforest of the ocean, and they really are. They're the most exuberant expressions of life on this planet. So anyway, I'm going to stop sharing there and hand it over to Dr. Gonthier, unless there are any questions or comments. Okay, take it away.
There was just one in the chat, Greg, and
okay,
Peter answered it. So you're good.
Okay.
Okay, well, thank you, Greg, for that. As Greg mentioned, I love going to Cozumel, as we would say in Spanish. To me, it's like going home. I grew up in the central part of Mexico in a town called San Luis Potosí, which is the capital city of the state by the same name. And I lived there until my father passed away at the age of 19. And I was already going to high school in Texas at a private boarding school, where I wound up meeting my wife, and She is from Honduras in Central America, Honduras. And we both continue to speak Spanish at home and randomly at different times. We don't mix the English and Spanish within words or sentences, but we often switch back and forth unconsciously. And so the first time I dove in Cozumel was in 1992, I believe, when I became certified. And ever since then, I love going back, and there's a possibility there that when we retire, we might decide to spend a good portion of the time in Cozumel. I have a favorite dive master that I dove with him in August of 2019. And since then, we have not been able to dive in Cozumel. We have supported him throughout this pandemic. And so we talk to each other every week for about an hour and carry on in Spanish, despite the fact that his English is good. But I have a video here that kind of introduces us to Cozumel. It's usually part of a larger video that runs about 20 minutes to introduce students to this field experience and try to recruit students for the trip itself. So this is a 3-minute video portion of that, and there is some sound that may play in the background here that I will try to mute a little bit, but it shows the island of Cozumel and the different dive sites. This is our hotel where we stay at. It has the largest pool on the island, and it's a very nice time for students to get into. This is our beach in front of the hotel, not much of a beach. These are the rooms. They're usually very nice and clean. This is a arrival day in which we do open waters, and you can see this is getting late in the afternoon. With the sun setting, we usually are able to accomplish the exercises, and then we go boat diving the following day. Here we're getting together, waiting to go out to dinner. It looks like this is probably our first dinner, and then this is a subsequent dinner later on. These are local places that I love to go to. This is a Sunday, so there's a big party in the square, and students are interacting with the locals. And this is now out on the boat as we're getting ready to go diving. Students are excited. Some of them are nervous. This is our halftime. We have a surface interval of about an hour to try to allow some gas to degas, some nitrogen to degas from our bodies. And this is some of the marine life that we see and some of the coral. Some fish. It's a queen angel fish. There's all kinds of beautiful fish. Little stingray. There's as Greg mentioned, there's a wide variety of fish. This is a dive on the wall. It becomes a deep dive, maximum depth of about 80 feet. This is now ending the dive up on the shore. Students really enjoy this time and they have a blast. We have a nice surface interval where we get some food. They interact with the crew. But they wind up seeing an awful lot. This is one of the larger fish known as a grouper, Nassau grouper, that we don't see very often anymore there. So some of these big predators seem to be disappearing. This unusual seahorse there, arrowhead crab. There are lots of those around, and sponges of all sorts, and Christmas tree worms, um, gray angel, and just all kinds of beautiful coral structures. That this is some shrimp and an anemone. And this is our dive master that time, and we're time to go home, to call the end of the dive. And so this was just a short video of the beginning of this longer video of about 20 minutes, and we may come back to it. But on March 2019, which was our last trip to Cozumel, there was a small group of students who came along with us. And we encountered this spotted eagle ray that was feeding. And there is some physics that we talk about these eagle rays, because there's a group of organisms, sharks, rays, and skates that are able to detect electric fields. And so they look for their prey with these sensors. And in the case of the eagle ray, he or she is looking for prey that is buried in the sand. And so this is a 3-minute video of, and this I would rate as my best video shot. And the video gets better, the pictures get better with time because the cameras are getting better. So this is a Hero 4 camera or Hero 6 camera that is taking 4K video now. And it's just a magnificent video that the spotted eagle ray allowed us to take of him or her. And so what's happening here is the organism is looking for the shells that are buried in the sand, and wherever senses an electrical pulse, she or he begins to dig there. You see these indentations of previous feeding episodes that have occurred. And there was several groups of divers, 2 groups of divers, I believe, were watching this. And we were just right up next to it. It's just something, I mean, sometimes we see these, but we see them swimming along the wall, and there's no chance to get up close to them. You have a little boxfish swimming behind this ray, trying to catch a scrap. You'll find that there's a lot of cracked shells along the bottom here that have been left over from previous. These animals are able to crack the shell open with their mouths. And it's just beautiful. It's a shallow dive and about 20 feet of water. You can see the surface there. And you see this guy in action. It's just amazing. You can see its eye there. And here it looks like he or she has really found something. You see some of the sand coming out of the exit area of the gills there. well, the gills are down here, I guess, but there's another. So we study this detection system called the Lorenzini cell in sharks that is able to detect electric fields and therefore also magnetic fields. And so probably it is through the same organ that they're able to detect the direction of the magnetic field, and therefore they have some sort of sense of direction. And this may be true also of other insects and birds as well, but it's a different mechanism, a different organ that is used for this. But finally, the spotted eagle ray has decided that that's enough show and tell, and decides to take off into the blue, as we might say. And so we leave that guy. The next thing I wanted to do is mainly talk about another interesting organism that we discovered. At least I didn't know anything about. And we did this from an experiment that we did in front of the hotel.
Hey, Pete,
And
there
that
was a-
is trying to measure the speed of sound in salt water.
There was a question there, Pete.
And so what we did is we obtained two hydrophones, which detect sound in the water. And then I used a voice recorder, which is just for recording speech in the classroom or something of that nature, but it records in stereo. And so each of these channels was a different hydrophone. And so we recorded the sound, and then the idea was to analyze the sound and be able to measure the time difference between the two hydrophones, and therefore decide on the speed of which sound is propagating. So here we had a sketch. We needed some measurements of the separation of the hydrophones, and there was a ladder on the dock. So this is a particular pier that we're using. We have a hydrophone at the end of the pier and one close to the ladder. This is our hotel, and this is where we catch the boat to go scuba diving early in the morning. This is where we do our open waters. And there's a ladder there which we hit with some piece of metal. I had a crescent wrench that I guess I used. This is our pier here. We're getting ready, waiting for the boat to come in this case. But this is a recording that we've made, and there is an audio to this that I will play, and it's only showing you the clicks. And I think you all were more or less able to hear that. And so then what I'm doing is displaying the amplitude of those signals by looking at them in a program that is a program that we use for research purposes and publication and so forth. But here we have one channel, and here we have another channel. So this is the hydrophone that is closest to the ladder, given that the signals are larger and they're coming slightly before. And this is the hydrophone that is further down. And this is in air. So the hydrophones were taken out of the water, and this was done in air. Then we use the same program to create what is called a sonogram. So on the x-axis here is time, and on the y-axis is frequency in hertz. And this is a distribution of frequencies as a function of time. And we can pick out where the most intense location of this is, which is shown in red here. And we can then find the time difference between these two. And we do several measurements, and we obtain an average, which is a little bit on the low side, with an uncertainty that we can obtain from these measurements and estimates of the uncertainties of individual measurements. You can see we're a little bit lower than what we expect, but the speed of sound also depends on humidity and temperature a bit. So I didn't take those things into consideration. The real experiment is when we submerged these. And this is where we encountered a crackling sound that sounded like electrical discharges. And we couldn't figure out what that was. And slowly, later, we were able to understand that this sound was produced by snapping shrimp. And then we started to study snapping shrimp, and we just fell in love with this critter that so we did the same experiment underwater, and so this is now the hydrophones in water, and we measure the time difference, and we get something reasonably close, a little bit on the low side, again very similar to what we found in air. Now, salinity might play a role in temperature and things like that, so who knows? So, but like I say, this measurement was key into allowing us to discover snapping shrimp. And these critters turn out to be very exciting in how they're able to use physics to focus energy, is the way I put it here. And here's our Latin name for these guys, Apheus heterocleus, or something of that nature. Greg is just so good at pronouncing these names, and he knows them all and just rattles them off, and it's not quite something I'm able to do, so I struggle with that. But in any case, snapping shrimp, we discovered them in 2008, which was, we made some hydrophone recordings. Then our first trip was in 2006 for the course. And so we've taught the course every other year, essentially, since then up until 2019. And these snapping shrimp were really discovered after World War II, when submarines were coming back into their home, and they were coming in to the I don't know, like the Bay Area, and all of a sudden they experienced this communication interference, and they couldn't figure out why it was coming from. And later they discovered that this interference was coming from snapping shrimp. And the sound is very sharp in time and therefore very broad in frequency, and therefore interferes with just about anything um, that is used for communication purposes. But we didn't know about them until this discovery. And this is the sound that we recorded in 2008. And another little sound recording in 2012. A little bit different. Here you can hear the surf going back and forth, but you hear these things like electrical discharges. And this is on a website of Dolphineer. And this is actually from the University of Twente in the Netherlands, where they did a study on snapping shrimp, and it's published, and actually published in very prestigious journals like Science and Nature. It's pretty awesome. So this is their sound. And this is just thousands of snapping shrimp doing their thing. Now, some of this can be used for communication, but some of it is actually a weapon for getting their food.
Far from being quiet, the ocean is filled with a cacophony of animal noise that can even disrupt the sonic transmissions of submarines. Most of the din is made by a surprisingly insignificant creature, the pistol shrimp. By snapping its claws, it can not only make communication sounds, but something far deadlier. These shrimps are its prey. It deals a knockout blow from a distance by using its claw as a sonic weapon. First, its claw is cocked like a pistol, then fired. The effect is literally stunning. As the claw snaps shut, it fires a blast of bubbles. Incredibly, as the bubbles collapse, they momentarily reach the temperature of the sun. This implosion causes a shock wave that stuns.
So obviously there's additional sounds that they've added there for effect purposes. Some of these people have them in aquariums, and so you can do some nice close-up photography of them. But this other group actually studied it in the laboratory and did some real scientific studies of it, and then proposed a rather complicated model for this bubble that is coming out, and it forms. And the question is, how does a bubble form? Well, the jet itself leads to a decrease in pressure surrounding the jet. And that decrease in pressure is below the vapor pressure of water. So it causes, if you will, the water to boil briefly, producing this bubble. And then this bubble finds itself in an environment of high pressure and rapidly collapses and just keeps on collapsing into a very, very tiny thing that finally explodes in a sense. And it's this collapse to something very, very small that increases its temperature. It's an adiabatic compression, and therefore the heat is contained within that bubble. And as they say, it reaches the temperature of the surface of the sun, about 5,000 Kelvin. And this is what they learned from their study. And here is a short video of their study in the lab in the Netherlands with a group from the Netherlands and Germany.
This is the sound of thousands of snapping shrimp. The snapping shrimp has one normal claw and one oversized so-called snapper claw, which can be closed with lightning speed and produces the snapping sound. The snap is used to communicate with other shrimp and is also used to stun or even kill prey animals. To study the sound production mechanism in snapping shrimp, the shrimp was tethered on a small platform and the sound signal was recorded with a hydrophone. The snapping shrimp was then tickled with a paintbrush and imaged with a high-speed digital video camera. Although it was always thought that the sound was produced by the two claw halves hitting each other, it is seen here that a large bubble is formed. It is the implosion of this cavitation bubble that produces the sound of snapping shrimp. The bubble is formed in a fast water jet squeezed out from between the claws. The water jet velocity is so high that the corresponding pressure drops below the vapor pressure of water, and a cavitation bubble is generated, which will initially grow in size, then it collapses violently. The high-speed video images and the sound recordings were correlated in time. In this side view, the claw is fully opened. At this position, the claw is fully closed, no sound, while the prominent snap coincides with the bubble collapse. We set up a model based on the Rayleigh-Placid equation to study the bubble dynamics. In this model, a Gaussian pressure drop was used to reproduce the bubble radius as a function of time, as also observed in the experimental high-speed video recordings. From the dynamics of the bubble, the sound pressure curve was calculated. The precursor signal before the loud snap, representing the bubble growth and shrinkage, are also in good agreement with the experimentally determined hydrophone signal. Now that you know that snapping shrimp use cavitation damage to catch their prey, for example, this small crab here, you better stay away from the acoustic weapon of snapping shrimp.
So they had a very complicated physics model that they came up with that described this, called the Rayleigh-Placid equation, that was known from previous studies of cavitation bubbles, because there is the possibility of generating these bubbles in the laboratory with radio frequency and sustaining the bubble and actually getting it to glow. And so this is referred to as sonoluminescence. And so the same thing applies to these bubbles here. And that's how this light that is given off comes from high temperatures that are initiating this emission. This is one of our recordings here where we have recorded the snapping shrimp, and this is the sonogram, and you can see the frequency spread is rather broad, but it's a very short time scale. And of course, we don't have very sophisticated equipment the way that they use to study this in the lab, but we can see why this would interfere with all kinds of frequencies that are used for communication purposes underwater. This is one of their publications. on snapping shrimp, and then of course they were able to come up with applying this model to the data and being able to determine some of these constants. And like I said, some of this work was published in Science and as well as in Nature. And so here's again some recordings and the snap really coming from the collapse of this cavitation bubble. Here's another video that was also shown in that study. This is very similar. Well, okay, here's some data associated with this collapsing of the cavitation bubble. We can see that the sound produced is very intense. The claw closes very rapidly, so you're talking about a plunger system that has a spring type that has a very large spring constant, and the velocity of the tip of the claw is rapid, producing this jet of the order of 30 meters per second. And so a cavitation forms. The bubble growth is in, almost 400 microseconds to a maximum 3.5 millimeters. So it's a small bubble, and then collapses in about the same time, 300 microseconds. A flash of light is then correlated with a peak, again, due to these very high temperatures. And this is the same sort of phenomena that one observes with propellers, where you have propellers producing these cavitation bubbles that then produce cavitation damage to the propeller. And you basically are melting the metal of the propeller away because of these high temperatures. And yet these critters, very small critters with a snap of their claw, are able to generate these and focus this energy for the purpose of capturing their prey. Just to give you an idea of how loud this sound of the snapping shrimp, of course, this is right at the cavitation bubble, the collapse of that bubble. But you can see pain is at 120 decibels. So if one of these occurred in our ear, we would feel it. Because 200 decibels is a very large intensity. This quickly dissipates in water, and so the recordings are not, in a sense, one has to correct for the distance away from the bubble itself. But in the laboratory, when they do these studies, they can determine the intensity. So what we did to kind of illustrate this or explain this is kind of have a plunger model. So it's like a syringe. And there is this area of the claw, and then you have this plunger coming in, which is the other side of the claw that is closing and then squirting this water through a narrow structure in the claw, and therefore coming out with a higher velocity. And what relates these areas to the velocity is the mass conservation or the continuity equation. And it's basically going to be the ratio of these areas that will determine the ratio of the velocities. In addition to this, we have Bernoulli's equation, which is something that one studies in an introductory physics course. And Bernoulli's equation is basically a conservation of energy. We have a kinetic term, we have a potential term, and we have a pressure term that we, in this case, is it's a energy density, if you wish. This is a density of the water, and the height at which this is occurring. And so if this is true at the beginning, it's also true at the end because this is a constant. So we have this scenario where we have this area 1, this velocity 1, we have this pressure 1, and the fluid then goes through at a higher velocity in a smaller area, therefore higher velocity, and decreases the pressure due to this Bernoulli effect. In this case, this is all happening at the same height, so there's no potential energy term to worry about. And so Bernoulli equation is well known in engineering. If you are wanting to get water up to the 10th floor, well, then you know what pressure you need at the ground floor in order to be able to do that. And basically here we have the relationship of the two pressures and the velocity. And we could say, well, maybe we're at a depth of 75 feet, and that's a little bit exaggerated. Normally you find these snapping shrimp in 20 feet of water or even less. But okay, I took this a rather drastic depth there. And so the idea then is that we have this jet of water, maybe of the order of 30 meters per second, that decreases the pressure around this jet. And there's some sort of nucleation that takes place, forming this little air bubble. Very small in size, as they indicated, something like 3 millimeters. It grows to that size, but maybe starts out to something of the order of 10 millimeters. And so I asked the question, well, how does the vapor pressure of water go with the temperature of water? And so basically, water begins to boil when the pressure is equal to the vapor pressure. And something around, so this is the vapor pressure of water. And you can see at 100 degrees centigrade, it boils at atmospheric pressure, which is 760 millimeters of mercury. And at 23, 24 degrees centigrade, this is marking the temperature of the water in the ocean. This is the vapor pressure that we need, which is about 24 millimeters of mercury, or in standard meters, kilograms, seconds, units is pascals. So it's 3,200 pascals. And so now we ask, okay, with this plunger model, we have 3.5 atmospheres here, and we produce a jet with this velocity. At what point do we reach the vapor pressure? And it's of the order of 26 meters per second. And this is assuming we're at a depth of 75 feet of water, which is rather extreme. So if you're in 20 feet, well, you're at down quite a bit, and this would get to the vapor pressure at a much lower velocity. But this illustrates that all of this is doable and understandable in terms of simple introductory physics. And so, as was mentioned, there is this flash of light, and so you have shrimpoluminescence, as this phenomena has been referred to. And in physics, sonoluminescence is well known and is associated with this cavitation bubbles that can be sustained in the lab and studied. And here in this case of the shrimp, we have this flash of light, which is very short duration and reaching very high temperatures compared to the surface of the sun. And so there was a very nice study, a theoretical model that was in place and able to explain things rather neatly. The model here has some oscillations that perhaps are not found in the lab, but they didn't say very much about that. So, but that's basically all I had. I will stop sharing and come back to you. And was wondering if there was, by any chance, some questions. And I see that Greg has answered some of these along the way.
There is one, Peter asking, I don't know if you saw the one about asking if your activity by the ray affects the ray's activity. And then also, what is the size of the snapping shrimp, if you have any idea?
Well, it's a couple of inches. The snapping shrimp are just, there's some that maybe grow bigger, but usually they're fairly small, of maybe a couple of inches. Our activity by the ray itself didn't seem to disturb it whatsoever. Now, I think the question may be, does my electric field affect its ability to sense the field of these organisms that are buried? And it's not clear to me that that would be the case. But I frankly do not really know the answer to that. It's a good question, though.
That I think the electric field attenuates with distance so effectively that we probably don't, that the electrical field that we and all living things generate is not strong enough to interfere over the greater distance. If we were really close, then it certainly would.
Yeah, it goes as the cube of the distance, so it really decreases quickly in intensity.
Hey, Pete, I thought we might show that video that we had professionally made for the last couple of minutes, if that's okay with you, and I have it on my computer.
Okay, so you can show that.
I think I can show that. Sometimes a professional photographer goes out with you on the boat, and then for $20 a piece or whatever, they show you the video, and you can buy it. And with that many of us there, it's just a few bucks from each of the people. So this actually shows a bunch of the organisms and a bunch of the students diving. And so I thought I might share that. And so let's try this. I hope the sound will come through as well.
Mommy love you, Daddy, all night long, all right now. Hey, hey, all right. See the girl with the diamond ring? She knows how to shake that thing, all right now, now. Hey, 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 hey. Tell your mama, tell your pa, I'm gonna send you back to Arkansas. Oh, yes, ma'am. You don't do right. Don't do right. When you see me in misery, come on, baby, see about me now, yeah. All right. All right. Oh, play it, boy. When you see me in misery, come on, baby, see about me now, yeah. Hey, hey, all right. See the girl with the red dress on, she can do the birdland all night long, yeah, yeah. What I say, all right. Well, tell me what I say, yeah. Tell me what I say right now. Tell me what I say. Tell me what I say right now. Tell me what I say. Tell me what I say, yeah. And I wanna know. And I wanna know right now. And I wanna know. And I wanna know right now, yeah. And I wanna know. And I wanna know, yeah. What? What's that? What's that? What time? All right. Oh. Oh. Ah, one more time. Say it one more time right now. Say it one more time now. Say it one more time, yeah. Say it one more time. Say it one more time, yeah. Oh. Ah, baby, feel so good. Baby, feel so good now, yeah. Ah, baby. Baby, feel so good, yeah. Baby, feel so good. Baby, feel so good, yeah. Oh.
So who can argue with Ray Charles? But I just want to say one other thing about the nature of the trip to Cozumel. Getting students off campus is just really, really important. They get to know each other, and we get to know them, and them us, in an informal way as people. And we talk about our families and all kinds of other stuff. And so these are really important experiences for college students. And Hope is trying to increase, as you probably know, increase the opportunities for students to do off-campus study. This is a short duration one, but it really makes a difference. We keep in touch with a lot of these students over the years, and they with one another, I think, too. So it's a real privilege for us, and I think for them too. There's some expense involved, and that's another thing that the college is trying to work on to make it more possible for more students to do off-campus study. So
Love that.
anyway, we're happy to entertain any other questions if you have.
Did get a couple in the chat here.
Okay.
Let's see. From Diana, over the years, have you noticed degradation of the reef? And if so, how would you characterize it? Is it irreversible? How serious?
Well, one of the things that we've seen from my perspective, which is shorter than Peter's, but in one of those, in the video, you saw a lionfish, and that's actually a Southeast Asian species that is invasive in the New World, that has just exploded since the 1980s, really. And in 2006, I think we saw none on Cozumel. By 2008, 2010, we saw several on every single dive. And now we hardly see any anymore, because the dive masters and sport divers have been employed to remove them. So everyone they see, they kill.
Oh.
And it's really helping, but that's probably only on the reefs that are frequented by sport divers there. So that's a change that I've noticed. I think the coral is actually probably recovering from the last hurricane damage. And Peter can speak to that better than I can, because he's been diving there virtually every year since, I don't know, sometime in the 90s probably.
Yeah, 1992 was my open waters there. Then I went back. I think the first group that I took was in 1994. Matt Nays was on that trip, and he sends us a postcard every Christmas.
Mm-hmm.
Matt Nays and
Krista,
yes, they got married afterwards, and now they have 3 kids. Yeah, on the lionfish, you also find the meat for sale at the market there in Cozumel. So it is kind of a bit of a delicacy for locals. A lot of the dive masters, if they find a big one, they will kill it and take it home for supper. They try to feed the smaller ones to green moray eels, and trying to get them to, because at first they've had no predators, and so that was a real problem. I believe it was in the fall of 2005 when the Hurricane Wilma remained over the area for 3 days as a Category 5 hurricane. And we were questioning whether we could go in 2006, which was our first trip of the class. And we did go, and we did notice that there was considerable damage to some of the coral, at least the fragile coral like elk horns, especially near the shallow areas. Now, a lot of sand was brought up onto the coral reef, and some of that winds up basically killing a lot of the coral. The other problem was was some new piers that went into Cozumel for the cruise ships. And one of these piers is close to Paradise Reef. And there was great concern for what would happen to this reef. And I remember diving this reef and as far back as 1994, and it was a gorgeous reef. And now it's pretty dismal. It's not, there are places, and that's usually where we dive now, but generally it was a large reef that was very pretty. But there's some loss of reef because of that. We do see the reef recovering. We're beginning to see some of the more fragile coral come back. We've had a couple of hurricanes go through there this season. Don't know what the damage is. I don't think they were fairly quick and went through rapidly, so I don't think it wasn't too bad. But yeah.
I would point out too, we haven't seen any damage there that we could attribute to things like coral bleaching that results from changes in temperature and water temperature, especially, and to some extent acidity of the water. Both of those are associated with climate change, and we haven't seen that on Cozumel yet, but that's coming in the future. And one of the major challenges for coral reef environments has to do with carbon dioxide in the atmosphere, which acidifies the ocean and also increases ocean temperature as a consequence of climate change. And coral bleaching, which is an expulsion of the symbiotic algae, photosynthetic algae that live within most of the corals, that kills a reef. And we're going to see that in the future in a greater, in a greater degree than we have thus far. But lots of reefs in the ocean, especially the Great Barrier Reef, is suffering from those kinds of damage right now.
Indeed. All right, I think that answered one of the other questions. And then, were dives always offshore in a boat, or were there interesting shore dives as well? And did you encounter any crocodiles?
The crocodiles there don't go into the seawater around the areas where we would be diving. They're only in really shallow areas in estuaries and so forth. But Cozumel is very safe. You do have to be a little bit concerned sometime just about, when you're with a group, it's not a problem. And it's such an area that has been developed for scuba diving. That's actually another reason why the reefs are in such good shape. All of Cozumel, all of that side of the island where sport divers go is protected as part of a national park. And so there's no extractive use like commercial fishing allowed there. And that's why we keep going back. It's well set up for diving. It's really efficient. We can make 3 dives per day, 2 in the morning, 1 in the afternoon. Virtually all of those by boat, but we do a night dive from shore as well. A lot of the students do. And we do our initial checkout dives from the shore right there at that hotel. That's one of the reasons why we use that hotel. We can get group rates that are very cheap, even though it's a really, really nice hotel. What we're doing is really efficiently done there, and that's why we keep going back. I've been diving at a couple of other places in the Caribbean, and the reefs are in much better shape in Cozumel, in part because it's protected by the government to support the economic impact of so many sport divers going there from all over the world. Fantastic. So Cozumel is the town is very safe. Yeah, we never have a concern there.
Well, that and the video look really good on this February cold day, that's for sure. Let me open it up. If anybody else wants to make a comment or question, you can unmute yourself. And then we will ask Bill to wrap it up with a thank you. Does anybody have anything else they want to ask or say? Okay.
This is Bill Wells again, and I would like to thank Greg and Peter for the really interesting physics. I'm a mechanical engineer by training, and I knew that cavitation was a problem for pumps and propellers and that kind of thing, not only a waste of efficiency, but also potential damage to the pump or the propeller itself. But I had no idea that shrimps could do it too.
That's
That's
awesome.
why we find it so fascinating too.
Yeah. Thank you very much.
Thank
you, all of
you.
you.
Let's
You're
give 'em a
very
hand.
welcome. It's always a pleasure.
It was awesome, you guys. We appreciate your time this morning so much.
Thanks. Us too.
Have a great
Yeah.
day, everyone.
Yeah, bye-bye, everybody. Stay
Bill.
safe.